Hi students, in the last class we discussed about the pigment activity, means the rate of absorption of light and the yield of carbohydrates, that is nothing but rate of photosynthesis, right? We discussed. Coming to the next topic is the photosynthetic pigments, right? Which are also called photosystems. We know about the photosystems. Two photosystems are present within higher plants. There is an PS1 and PS2, and right, these are the photosynthetic units, which are also called photosystems, and other name is also there, that one is pigment systems. Other name is the pigment systems, right? So this is about the photosynthetic units or the photosystems or the pigment systems, which are nothing but PS1 and PS2. When we discussed about the chloroplast, that time we discussed about there is a thylakoid membrane. Within the thylakoid membrane, two types of pigments are there, PS1 pigment and other one is called as PS2 pigment, right? PS1 is nothing but photosynthetic, right? See, this one is called as photosystem 1, right? Other one is called as photosystem 2. PS1 photosystem 1, PS2 photosystem 2. So today we are going to discuss about the components which are present in PS1 and components which are present in PS2, right? So in that case, the very first one is the PS1, right? See, we know the PS1 and PS2 major function is the light energy is converted into chemical energy, right? Here the PS1, LSC1. See, within the PS1, majorly two components are present. There is a center. Okay, there is a center pigment is there, right? The center pigment is called as reaction center. That reaction center covered by many number of pigments. That reaction center covered by the many number of pigments or many types of the pigments. See, the center one is called as reaction center, right? Remaining other pigments are called as light harvesting complex. The remaining other pigments are called as light harvesting complex. These are also called antenna 1 complex. Okay, in the last class we discussed about there is an antenna molecules are there, right? Antenna complexes are there. So where the center, right, there is a main reaction point is present. That main reaction point is called as reaction center. Other than reaction center, remaining other pigments also present, right? These are called as light harvesting complexes or antenna molecules or antenna complexes. So LHC1, right, pigments, there is in chlorophyll A 80, there is in chlorophyll A 80 molecules are there, there is in chlorophyll B 20 molecules are there. So there is in we know that ratio 4:1. You know the ratio, that is the 4:1. See, at the center chlorophyll A is there, and the chlorophyll A is surrounded by the remaining different types of pigments. Chlorophyll A is present, chlorophyll B is present, carotenoids are present, and the xanthophylls are present. So if we discuss about the carotenoids, we know about the carotenoids which are having the orange color, right? So within the LHC1, more amount of carotenoids are present and less amount of xanthophylls are present. We know about the xanthophylls also, which also exhibit the yellow color. So all these components are present within the PS1, right? Other one is the reaction center. See, this is a very important center point. There is a specialized modified chlorophyll A molecule is present. There is a specialized modified chlorophyll A molecule is present, right? That one is majorly involved in the conversion process. The conversion is nothing but the radiant energy, that is nothing but light energy. Light energy is converted into chemical energy. Light energy is converted into chemical energy. Absorbs the longer wavelength more than 680 nanometers. So the PS1 absorbs the light energy, the wavelength more than 680 nanometers. Okay, 680 nanometers. So this one absorbs the longer wavelength. Right? So other one is the, this is about the PS1 in the same manner. Okay, there is a PS2 is also present, and I like to discuss about one important point. I said the reaction center contains chlorophyll A, and the accessory pigments also contains. These are nothing but accessory pigments. These are also called light harvesting complexes. Accessory pigments also contains the chlorophyll A. So what I'm telling here, accessory pigments also contains chlorophyll A, but the reaction center also contains the chlorophyll A. But the chlorophyll A is very, okay, whatever that reaction center contains, chlorophyll A is present. That one is very special. That one is very special. Okay, the special modified chlorophyll A molecules present at the center, right? That is about the PS1. That is about the PS1. And next one we will discuss about the PS2. Within that PS2, there is an LHC2 is present, right? One is the LHC2, right? Other one is nothing but reaction center same, but little difference is the reaction center. Other one is the reaction center. So LHC2 here there is an pigments are present. There is a pigments are present. Remember, I forgot to tell one important point. Along with these pigments, some of the proteins are also present. Along with these pigments, some of the proteins are also present in both the cases. LSC1, there is pigments are there. These are the pigments. Along with the pigments, proteins are also present. Here also, proteins are also present. In both the cases, proteins are common. Proteins are common, but the composition is altered. The composition is altered. So within the pigments, we know there is an chlorophyll A is present, right? The chlorophyll A, there is an 125, and the chlorophyll B, that one is also 125. So the ratio is here 1:1 ratio. The ratio is here 1:1 ratio. And next one is the carotenoids. Next one is the carotenoids. Here less amount of carotenoids are present and more amount of xanthophylls are present. More amount of xanthophylls are present. Simple, here composition is quite different from the LSC1. Here the pigments are present, proteins also present. Here also pigments are present, proteins also present. But the composition is different. Chlorophyll A 125, chlorophyll B 125. Ratio is 1:1 ratio. Carotenoids, okay, less number of carotenoids are there and more number of xanthophylls are present. More number of xanthophylls are present. Here reaction center. Coming to the reaction center, special chlorophyll A molecule, same. Special chlorophyll A molecule is present, right? So the radiant energy, converted into chemical energy. The radiant energy converted into chemical energy. Very simple. Here within the PS2 reaction center also same thing. There is no change here. There is a special chlorophyll A molecules are present. The radiant energy is converted into chemical energy. But the difference is here. This one absorbs the this one absorbs the wavelength less than 680 nanometers. Here more than 680 nanometers. This one is less than 680 nanometers. Here pigment is 700. Here pigment is 700. Here pigment is the 600. Here pigment is the 600. I don't have space here, that's why I mentioned here, right? There is a pigment 680. The pigment is here 680. So here within that PS2 there is an LSC2 and pigments are there, 125 chlorophyll A and chlorophyll B 125, right? 1:1 ratio. Carotenoids are less and more xanthophylls are present and along with these structures proteins also present, right? This is about LAC2 and the reaction center, special chlorophyll A molecules, right? Special chlorophyll A molecule is present and it's involved in the conversion of radiant energy into the chemical energy. Here the major pigment is pigment 680. It absorbs the wavelength less than 680 nanometers. It absorbs the light. The wavelength is less than 680 nanometers. Here more than 680. Here more than 680. Here less than 680. That is the difference between the absorption difference between PS1 and the PS2. Next one we'll discuss about the accessory pigments and how the transportation takes place, right? We'll discuss. First of all, we discuss about the composition and the functions of LS, PS1 and the PS2 also, right? Right, this is the arrangement process. Exactly this is the arrangement within that photosynthetic units. We are already known about the composition. See, within that case, how the arrangement takes place. See, at the center point, there is an reaction center is present. This is the reaction center. The reaction center, that is nothing but chlorophyll A. The reaction center is nothing but the chlorophyll A, right? Remember, this is a very special and modified chlorophyll A molecule. Finally, whatever that accessory pigments are present, the accessory pigments contains energy is grabbed by the chlorophyll A or taken by the chlorophyll A, right? So here there is in chlorophyll B. See the next row, that is the chlorophyll B. Next one is the again chlorophyll A. These are normal chlorophyll A. These are not special. These are not reaction centers. Right? There is in chlorophyll A and carotenoids are there. Xanthophylls are there. Or suppose this is the total antenna structure. Within the total antenna structures, okay, these pigments are arranged. These pigments are arranged. Okay, so this is about the arrangement, and this one is the major site for the reaction process, and these are called as accessory pigments. These are also called LIC complexes, light harvesting complexes, which are also called shield pigments. Which are also called shield pigments. Why these are called shield pigments? Which are involved in the protection. Okay, especially it prevents the photo-oxidation. Prevents the photo-oxidation. Other one is which prevents the solarization. So which prevents that photo-oxidation process, which prevents that solarization, right? So that is about that the pigments which are involved, right? And this is the reaction center. Once that reaction center absorbs the light energy, okay, this is the normal state and this is the excited state. This is the normal ground state. We know electrons ground level state is there and excited state is there. So this is one of the excited state. Again, these electrons are transferred to electron acceptor. The remaining pigments also there. We will discuss. PS1 pigments are different from PS2 pigments. We will discuss. Some of the electron accepting complexes also present, which accepts the electrons. So this is about the photosynthetic unit. Right? See, here we have to discuss about the LIC1, LIC2 and the functions also, right? Here there is an reaction centers. Reaction centers are there, right? Within that case, the very first one is PS1, right? Other one is PS2, right? See, the PS1, it's having that pigment 700. It absorbs light energy. And PS2. Here having that pigment is the 680. This one is the 700. This one is 680. This is the pigment 680. Right? It absorbs the light energy. light energy, right? So that is about that high level absorption process and the low level absorption. Means here more than 680 nanometers wavelength light is absorbed, here less than 680 nanometers light absorption takes place, and both are commonly having that complexes are called as LHC1. Here this one having the LHC1 complex and this one having LHC2 complex. So and which are also having proteins, LHC1, LHC2 and proteins. So what are the functions are performed by the reaction centers? Remember, the reaction centers are majorly involved in the conversion process. See the PS1 and PS2 which are involved in the ATP synthesis process, which are involved in the NADPH2 synthesis process, NADPH2 synthesis process, and evolution of oxygen. These are the functions of PS1 and PS2. There is an ATP synthesis takes place, NADPH2 synthesis takes place, and oxygen evolving, means oxygen evolution is also takes place here. That is about the reaction centers, right? We'll discuss about that how the H plus ions are pumped, means how the electrons are transported, how the H plus ions are pumped, how the ATP synthesis takes place. We'll discuss now, right? Yeah, this is the main concept for the light energy is converted into chemical energy. In that case, there is a light reaction is there. This one is also called photochemical phase. See, photochemical phase is nothing but the radiant energy or light energy is converted into chemical energy. Here there is a light absorption process takes place. That absorption is supporting for the photolysis of water. We know this is the one of the thylakoid membrane. This structure is called as inside. This structure is called as lumen, and this part is called as stroma, right? Inside the lumen there is a complex is there, right? Oxygen evolution complex is there, especially that complex involved in the photolysis of water. So during the photolysis of water, the H2O is converted into 2 H plus ions, especially one water molecule I am talking. There is an oxygen molecule and the electrons are released. So here, see, there is an 2H2O, that is nothing but 4H plus ions and O2 production. 4H plus ions and the O2 production. So here what I am telling, inside the lumen, there is a photolysis of water takes place. So here we are discussing about one water molecule. So there is a 2H plus ions are produced and 2 electrons are produced. 2 H plus ions are produced and 2 electrons are produced. So once the light energy is utilized, 2 H plus ions are formed inside the lumen and 2 electrons are produced. These electrons are transported through the electron transport chain. So there is an electron transport chain is there. Here there is an electron transport chain is there. That electron transport chain involved in the transfer of electrons. Finally, it synthesizes the NADP is converted into NADPH2. NADP is converted into NADPH2. So there is an NADPH2 production takes place. So here the use of electron transport chain, it transports the electrons that time from stroma to lumen. This is the stroma, right? From stroma to lumen, 4H plus ions are transported. 4H plus ions are transported from stroma to lumen. 4H plus ions are transported, right? So release electrons and 2H plus ions when the photolysis takes place. After that, when these 2 electrons are involved in electron transport chain, that time 4H plus ions are transported from stroma to the lumen. Total H plus ions are 6H plus ions. So end of the electron transport chain, there is an NADPH2 production takes place. That is nothing but NADP is converted into NADPH2. Then totally here 6 H plus ions are present inside the lumen, and these H plus ions are transported through only ATP synthase enzyme. This is the ATP synthase enzyme. That ATP synthase enzyme involved in the pumping of H plus ions. From lumen to the stroma. from lumen to the stroma, because inside the lumen more H plus ion concentration is present. Then for the balancing purpose, these H plus ions are transported through only the ATP synthase enzyme. Then here these H plus ions are transported from lumen to the stroma, right? See, that energy is utilized. Then in that case, the ADP plus PI is converted into ATP. The ADP plus PI is converted into ATP. Remember, the mitochondrial photosynthesis is different from chloroplast photosynthesis process. In mitochondria, whatever that energy is produced, that energy utilized for the cell functions. But in chloroplast, whatever that ATP is produced, that ATP is only utilized for the photosynthesis process. And remember, in mitochondria, 2 H plus ions is equals to the 1 ATP, but in photosynthesis process, 3 H plus ions are equals to the 1 ATP. 3 H plus ions are equals to the 1 ATP. So here finally 6 H plus ions are present inside the lumen, and these 6 H plus ions are transported through the ATP synthase enzyme. Finally it produces the 2 ATP molecules. So initially H plus ions are transported from stroma to the lumen by the electron transport chain. Once the high concentration of H plus ions are present here, right? In that case, here these are transported through the ATP synthase enzyme, right? So after that, which formed from the lumen to the stroma through the ATP synthase enzyme, then the ATP synthase enzyme involved in the synthesis of ATP, right? So this is a just peripheral explanation for the ATP synthesis and then ADPH2 and evolution of oxygen. Okay, there is an in detail explanation is there. We'll discuss in the next class, right? For the detailed explanation, how the electrons are transported, how the H plus ions are pumped, and where the H plus ions are pumped, right? And at what, at which point there is an NADPH2 synthesis takes place, right? So we'll discuss based on all these parameters in the next class.